Hello friends, Ace Bottain, next episode. In this episode, we'll be discussing about molecular basis of inheritance 10th one. So what all things we'll be discussing in this episode? For that I will share the screen so it will be convenient for you. I think screen came, so we can proceed. So in this episode, molecular basis of inheritance 10th one, mainly we'll be discussing Human Genome Project. Short form is HGP. So next episode, we'll be doing fingerprinting, last topic of this molecular basis of inheritance. So we can proceed to second page. An introduction will be there, then Human Genome Project and 5 theory question, 5 NEET model question with the key. So we can proceed with the introduction. Say Human Genome Project, what it means? It is the totality of humans. Totally humans will be analyzed genetically. And there the analysis will be on 20,000 to 25,000 approximately genes. And previous estimate, it was 80,000 or 1 lakh up to 1,40,000 genes in human DNA. But presently it is around 20,000 to 25,000 genes. More specifically, around 30,000 genes will be analyzed. So entire humans will be, the features will be led. That is Human Genome Project. But in fingerprinting, among the total humans, how we can identify individual. And remember, even though 99.9% of the base sequence among humans are same, that is what analyzed in Human Genome Project. Fingerprinting, the difference of each individual will be analyzed. And that fingerprinting will be doing in next episode. So I think you got what is Human Genome Project, what is fingerprinting. And remember, in both cases, Human Genome Project as well as fingerprinting, genes and DNA are the base material. So many steps will be common and in between, somewhere some deviation will be there between these two because aims will be different. So we can now concentrate on Human Genome Project. Human Genome Project, HGP, what is it? Why is it? Because when we study anything, we should know for what, why it is introduced. See, HGP is a mega project and it started in 1990. It was a 13-year project, was supposed to complete in 2003 and they almost completed in 2003, but chromosome 1 analysis was over in 2006. So that is what they suggest. It is 13-year project, 1990 to 2003. Many nations and organizations participated in it. Some of the agencies and nations include mainly US Department of Energy, USDE, National Institute of Health, NIH, later Wellcome Trust of UK, contributions from the nations like Japan, France, Germany, China, others. And chromosome number 1 analysis was over in 2006, even though it was a 13-year project. In India, there is a famous institute, CCMB, Center for Cellular and Molecular Biology, Hyderabad, doing a lot of work in fingerprinting and genetic related areas, CCMB. Next episode, we can discuss about it. Now, cost and magnitude of HGP. Cost of the sequencing for one base pair at that time was $3. Approximate number of base pair, it is 3 into 10 raised to 9 base pair. So cost estimation is around $9 billion. Remember at that time. And huge need for scientists, technicians, other staffs, and machineries. So it's a Herculean task. It is a huge project, but it is useful for entire human beings. Now, regarding storage of this data needs, because huge data will be there. When we tell in a theoretical manner, suppose there are 3,300 books, where in each book have 1,000 pages, and each page can accommodate 1,000 typed letters. That much books are required and that much quantity is needed. Then this data preservation, the retrieval, analysis, need computer system. So these all led a new era in biology. That itself is a famous branch, bioinformatics. That is how it started. I think first this page is clear to all of you. We can proceed to next page. In HGP, G stands for genome. So what is meant by genome? Genome basically means haploid set of chromosomes in a gamete or microorganism or in each cell of a multicellular organism, haploid set of chromosomes. Now, what are the goals of HGP? It's a theoretical aspect. Many aims are there, many goals are there. See, identify all the approximately 20,000 to 25,000, almost coming around 30,000 genes in human DNA, valid genes, and determine the sequences of the 3 billion chemical base pairs that make human DNA. Then store this information in database and only we can analyze it. Improve tools for data analysis. Transfer the related technology to other sectors such as industries. Address the ethical, legal, social issue, which is otherwise called LC, that may arise from the project. So these are some basic aims and goals of Human Genome Project. I think page is clear. We can proceed to next one. See, there are wide-varying applications for this HGP. Some of them include diagnosis, treatment, prevention of disorders, understanding human biology, then learning about non-human organism, learning, here one N, I think extra. So learning about non-human organisms, then healthcare, agriculture, energy, production, environmental remediation, some applications. Now, some model organisms used may not be human, non-human organism, like bacteria. Here you can find bacteria, then yeast, then comes Sinorhabditis elegans, the famous organism. It is a free-living, non-pathogenic nematode. Drosophila, plants like rice, Arabidopsis, and the famous plant. And this all organism, as a model organism, non-human organisms have been sequenced. Arabidopsis thaliana, one of the smallest genome among plants. And this is the first plant where genome were sequenced. So many importance are there for this Arabidopsis thaliana. Now comes methodologies. NCRT-wise are basically two methods are available. One is express sequence tag. Second is sequence annotation. So first we can proceed with the express sequence tag. What is it? Why that name, all this? So when we come to express sequence tag methodology of Human Genome Project, one method, here the procedure is identifying all the genes that are expressed as RNA. That is how the term expressed. Referred to as express sequence tag, short form is EST. It's NCRT representation, page number 103. Now comes sequence annotation. In sequence annotation, it is a blind approach of simply sequencing whole set of genome that contain all the coding and non-coding sequence. All will be analyzed. But remember, later, assigning different regions in the sequences with function is identified. That is sequence annotation, SA. So express sequence tag, all the genes that express as RNA. Sequence annotation, simply sequencing whole set of genome, finally required only will be taken. I think EST is an essay. It is clear. Theory question, NEET exam question. Now comes basic steps in HGP. So it is whether it is HGP, whether it is fingerprinting, basic material is DNA. So for sequencing, total DNA from a cell is isolated. It is called extraction. Total DNA. So that will be converted into random fragments because otherwise entire DNA not easy. It is a long polymer. So cut into fragments, convert into random fragments. After that, it is cloned into suitable vectors. Cloning resulted into amplification. Amplification will be remembering in genetic engineering side making many copies. So result into amplification of each piece of DNA by which sequencing will be AC. And commonly used hosts were bacteria, yeast, and the vectors were called as BAC, bacterial artificial chromosome, YAC, yeast artificial chromosome. Now, these fragments were sequenced with the automated DNA sequencers. That part is the difficult part now, but machines are available. DNA sequencers that worked on the principle of a method developed by the famous scientist Frederick Sanger. I think up to that it is clear. Now we can proceed to next page. NCRT diagram we can find, they are represented in a total manner. So we can start with the famous scientist Frederick Sanger, English biochemist, born on 13/8/1918, died on 19/11/2013. So birth and death. Now, Nobel Prize. 2 times he received Nobel Prize. A very rare chance to receive Nobel Prize in twice. Very rarely scientists we can observe in their field. 2 times getting Nobel Prize. The scientist got the Nobel Prize twice, and both in chemistry, but connected with biology. In 1958, first Nobel Prize, that is in chemistry for structure of insulin, the famous chemical. And second Nobel Prize in 1980 in chemistry for base sequencing of DNA or nucleic acid. That's what that sequencer developed. Such a famous scientist, Frederick Sanchez. So, using a sequencer, analysis will be done. That original part not directly mentioned in NCERT. It is a difficult one still. See, these sequences were then arranged based on some overlapping regions present in them. This required generation of overlapping fragments were sequencing. Required for sequencing. Alignments of these sequences was humanly not possible because that much complexity will be there. Therefore, specialized computer-based programs were developed. Here you can find they shown that computer system, etc. These sequences were subsequently annotated and were assigned to each chromosome. That is how the practical side. The sequence of chromosome 1 was completed in May 2006. That is why 13-year project, 1990, 2003, it is over. But the chromosome 1 fully completed in May 2006. Still we'll be telling it is 13-year project. This was the last of 24 human chromosomes. So many will be having doubt how it is 24. Humans will be 23. Because here 22 autosome. And males you can find X and Y. So that has to be taken separately. So 22 plus X, 23 plus Y, 24. That is how it has been become 24 and has to be sequenced. Another challenging task were assigning the genetic and physical maps on the genome. This was generated using information of polymorphism and micro satellite of etc. That we'll be learning in DNA fingerprinting. So it's directly linked. DNA is the material. Now comes famous NCERT data where usually questions will be asked. Some famous features of human genome. Here, this 1 or 2 questions from this aspect itself they'll be asking. So genome, mainly data, numerals. So genome contains 3164.7 million base pair, human genome. Average gene consists of 3000 bases, usual question. Sizes can greatly vary. Large non-human genome being dystrophin. That is why I underlined it. Dystrophin at 2.5 or 2.4 million bases. Total number of genes estimated at 30,000. That is why I told you in the beginning. 25,000. Originally it is around 30,000. Much lower than the previous estimate of 80,000 or 1,40,000. That is initial belief. Almost all 99.9% nucleotide bases are exactly same in all people. The remaining 0.1 will be analyzed in fingerprinting. Functions are unknown for over 50%. So our database question, very common important question. So functions are unknown for over 50% of the discovered gene. We don't know what is the function. Now next, again data continuing, less than 2% repeated question. Less than 2% of genome codes for proteins because NCERT, we cannot avoid it. Then repeated sequences make up very large portion of the human genome. That's all coming under fingerprinting. Repeated sequences are stretches of DNA sequences that are repeated many times, sometimes hundreds to 1,000 times. And they are thought to have no direct coding function, but they shed light. So even they have used, shed light on chromosome structure, dynamics, and evolution. Now, chromosome 1, another repeated question. 1 has most gene, 2968, and Y has the fewest, 231. Scientists have identified about 1.4 million locations where single base DNA difference, that is important data and expansion, SNPs, single nucleotide polymorphism, pronounced as SNPs, occurs in humans. And this information promised to revolutionize the process of finding chromosomal locations for disease-associated sequence and tracing human history. See the important SNPs. Now, next page. Application for future challenges. So this human genome project can help in understanding of biological system. Similarly, different disciplines in both public and private sector worldwide can be utilized by using Human Genome Project. Biological research, a big area. Then tissue or organ or tumor connected with the entire humans. All this can be worked by this Human Genome Project. And finally, chemistry of life connected with the humans. Now we can find after this 5 theory question, 5 NEET model question with the key. Hoping that it is clear to all of you. So what all things we learned in this episode? That is molecular basis of inheritance, 10th one. 10th one, we started with the introduction. This is, DNA. This is Human Genome Project representation. Then we entered into topics. What is the difference between Human Genome Project and the fingerprinting and number of genes? And both cases, DNA will be analyzed, base material. Then Human Genome Project, what it is or why is it 1919 13-year project completed in 2003 but extended. Then which all agencies involved and what is importance of CCMB in India, cost and magnitude of Human Genome Project, bioinformatics. Then G stands for genome, haploid set of chromosome. Then goals of HGP, almost 6 important aims, including LC. After that, these experiments, this wide-varying experiments done in non-human organism initially, and that include model organisms, bacteria, then fungus, yeast, fungus you can find. Then after that, Caenorhabditis, then Drosophila, rice, as well as Arabidopsis thaliana, importance of Arabidopsis thaliana as the plant, as well as Caenorhabditis elegans. Then 2 methodologies, express sequence tag and sequence annotation. What will be done in ESTs? So all the genes that expressed as RNA will be analyzed. Sequence annotation, analyzing the whole set of genome that contain all the coding and non-coding sequence, later assigning different regions in the sequence with a function, SC. Then basic steps, extraction, converting into fragments, plant, then BAC, YAC, then fragments, sequence using automated DNA sequencers developed by Frederick Sanger's method. Then famous scientist Frederick Sanger, British, birth and death, 1918 and 2013, 2 times a Nobel Prize. One is in chemistry, insulin, other is in base sequence, nucleic acid. Then this is the methodology or diagram in NCERT. Then what is the idea behind it? Then after that, sequence of chromosome number 1 completed only in May 2006, last of the 24 human chromosome, 22 plus XY. Then salient features of human genome, that is most important item. When we analyze the previous question papers of NEET, from here 2 questions normally they are asking. Some data are given. Then after that, 1 more set. Again, important area. This 9 points, very, very common for NEET exam, important for NEET exam. Important items I underlined there, so it will be easy for you. Then application and future challenges for this project. Then after this, as usual, 5 theory question, 5 NEET model question with the key. If you like this episode, subscribe all, like it, share it, comment on it. We can meet in the next episode for the last part of this molecular basis of inheritance called as fingerprinting. Thank you. We can wind up. Take care.